Hi guys, welcome to another Biology with Miss Cherry. As you can see today we are looking at insects. So before we begin, as per usual, please have a go at pausing the video and attempting these questions. So how does surface area to volume ratio affect diffusion? How do insects get gases into their bodies? And how do insects move? You might not know these, you might know these. Either way, I'd like you to have an educated guess. Okay, do your best and we'll go through those in a second. So pause now and play again when you're ready. So the first thing, or the first question anyway, is reflecting back on what we did last lesson. So the bigger the surface area to volume ratio, that is the surface area of an organism compared to its 3D volume, the amount of space it takes up, the more likely it is to transfer substances by diffusion. It means that there's more surface compared to how spacious it is, and therefore you're going to be able to move substances such as oxygen nice and quickly into the cells in the centre of the organism. Now, how do insects get gases into their bodies? Well, if you didn't know, this is what we're going to be looking at today. But you might know, and you might recall from last lesson, I mentioned about the tracheoles and the spiracles. If you didn't know and you said something about the lungs, that's a great guess, but not quite right for insects. They're too small, okay? They're not going to be able to fit a set of lungs in there. If you ever stepped on a bug, you know they're squishy. There's no organs in there. There's just like weird tubes and then a sort of pumpy heart thing. And how do insects move? Well, obviously they've got their legs and they've got their wings, but some of you might have also mentioned about contractions of the abdomen, and these are the way that these insects, and especially things like locusts, are going to help pump gases out and get gases back in again. Okay, so let's get started. Now you might be thinking, hey, this is lesson 3.3, we've only done lesson 1. Yeah, that's true. Normally I would put a practical in about surface area and volume ratio and do a practical when we get back to school. We can have a go. But this lesson is looking at unicellular organisms just briefly. We're not going to spend too much time on it. And then having a look at how insects do gas exchange. So the video on YouTube will be lesson 2 for module 3. Whereas in like the way that I would normally teach it is lesson 3 for module 3. So to begin with, have a look at this picture. We've got a unicellular organism, some form of bacterium. How would organisms like that single-celled bacterium exchange gases, and why would they do it in that way? Think back to the starter. We're going to recap this over and over and over and over and over until you guys have got it in your little noggins. Okay, pause, answer the question, and then play again in a second. So our unicellular organisms, like this bacterium, are small, and you should have been able to recognise that they have therefore got a large surface area to volume ratio, and therefore they are able to transfer substances by diffusion. Okay, even if we've got a cell wall there, you might remember back to the prokaryotic cells lesson that you did in module 2, there's no barrier to diffusion. Okay, these organisms are perfectly happy taking substances in and releasing substances via diffusion. Conversely, insects have got a very complex gas exchange system, and it's not too dissimilar to that seen in humans. Okay, this is a process of evolution giving them this very specialised system. If we take a look at the picture at the bottom, from the outside we have got pores called spiracles. And actually, these pores sort of look like the guard cells that surround the stomata that are found on the underside of leaves and plants that undergo transpiration. So as you can see, we have got sort of a door-shaped kind of mechanism. So they can be closed, or these spiracles can be closed, or the sort of sideways bits can withdraw a little bit, and then we've got an open pore. And this is where we're going to get gases moving in and out of our insect. Following on from the spiracles, we've got the trachea, similar to the trachea that we've got, and then they separate themselves down into tracheoles. Okay, imagine it very similar to the bronchi and the bronchioles in humans. Now these tracheoles, the parts at the end of the trachea that we've seen, permeate into body fluid. So if you remember last lesson, we had a talk about tissue fluid and how that helps with diffusion. Insects are so small, they don't have a complicated circulatory system. Instead, all we're going to get at these tracheal points is the movement of substances in and out of that body fluid by diffusion again. Now, much like in us, we've got structures that help support the respiratory system, the breathing tubes, things like that. And the trachea are strengthened by rings. We don't need to know what they're made of, but they keep the tubes open. Okay, so they keep those the lumen nice and wide so we can get that gas exchange going on. And finally, as we've said, the spiracles are where we're going to meet the outside world. And this is where external factors, things like wind and stuff like that, can start to have an effect on the rate of gas exchange. So as you can see from this number down in the bottom corner, this is not a very complicated lesson. We've only got 17 slides. So we're going to take it slow, and if you get confused with anything, please just ask a question. I would much prefer to hear from you than not. So let's have a think before we move on any further. Why do you think it's really useful for those tracheoles, the bits that we've highlighted in blue time and time again, are in contact with the body fluid? So you can pause now, spend a little bit of time. Yeah, think about it. Why is it useful that these tracheoles are in contact with the body tissues in that body fluid? Why is it good for them? So pause now and play again when you're ready. Now, the first thing that is important about this direct contact is that if you imagine my oxygen, I'm going to write in this like browny purple colour, the oxygen comes in through the spiracles, down the trachea and into the tracheoles, and it's able to have a direct contact to the tissues, to that tissue fluid. Okay, remember, atmospheric air has got other things in it. It's got some nitrogen, we've got some carbon dioxide in there as well. But it means that the oxygen that we need from that atmospheric air is able to pass straight into tissues that need it. And as well as this, we get a really short diffusion pathway. And that gives us quicker diffusion to tissues that need it. It's going to make... And likewise, if we're getting oxygen to tissues that need it, it means that we can also remove waste products a lot more efficiently as well. Don't forget, we're talking about putting in nutrients, but also removing the stuff that could be harmful. Now here we've got a gas transfer gradient flow chart diagram thing. Okay, we're going to take it from the top in the top left corner with the green words. So the oxygen concentration is highest in the atmospheric air and lowest at the respiring tissues. So if we take our diagram, I'm going to do a horrible sketch again. We've got the spiracles there, the trachea, moving down into the tracheoles. I'm just going to do one side of it just to make it a little bit easier. Okay, and then we've got the body tissues, or the body fluid, bathing the ends of those tracheoles in. And if we write concentration of oxygen, or O2, as a size related to the concentration, so big oxygen outside and small oxygen in the respiring tissues, therefore we can see that we've got a nice concentration gradient ready for the oxygen to take. Okay, so it's diffusing from high concentration in the atmospheric air to low concentrations in the body tissues, and then we end up with an equilibrium situation. Remember that this oxygen is then going to diffuse away into tissues to be used for things like respiration. So we're maintaining our concentration gradient. Okay. And likewise, when we're talking about gas transfer, we've got to think about the waste products. We can't forget those. So we've got concentration highest in the body tissues. So diagram time again. Spiracles, trachea, tracheoles. I'll go down here this time. And we've got more carbon dioxide, I'll do it in green, in the body tissues than in the atmospheric air. So again, we get diffusion, but in the opposite direction until an equilibrium situation is reached. And again, we've got to remember that that carbon dioxide can diffuse away, which is going to help maintain our concentration gradient. But at the same time, we've got to think about other factors that might help keep that gradient there as well. So we might have wind flow, or we might have pressure differences, which we're going to take a look at in a second. Okay, now this is a very, very quick process. When we're moving our gases in air, we've got much, much faster, more kinetic energy than if we were to move slowly through a liquid. So the diffusion of, say, oxygen through the body fluid is a lot slower than this process of moving carbon dioxide out of the organism's body. And as we can see, we're still talking about those gradients. So it's important to remember in an exam situation that you should be mentioning each part of the insect's respiratory system. So spiracles have taken the atmospheric air, they travel down the trachea to the tracheoles to the body tissues. Please don't miss any bits out. There's only a few things to remember, and one of those is air. You should be able to do that, I'm sure. But now we're going to take a look at something a little bit different. You might have seen this instrument before. It's called an accordion, and it's played by drawing the sides of the instrument out and then pushing them together and forcing air through the holes that you can see at the front. How do you think an insect is like an accordion? You can pause, you can have some thoughts. If we were together, we'd have a discussion, but we aren't, and you guys don't talk to me. So, make some notes if you fancy it. Now, the way an insect is like an accordion is because its body is able to wiggle around. Okay, it's generally made of small shell-like structures. It's exoskeleton that sort of slide over each other. Because we've got these segmented plates, they're able to move around and contract and relax. So they can sort of scoop up the abdomen shape. So we get a small volume, but a high pressure. And then they can relax where they become quite long again. And then we've got a larger volume, but a low pressure. And in doing this, we can end up forcing the air in and out of our body. Okay, the pressure inside the trachea changes. And this is going to be a process of mass transport. We're transporting lots of gases at one time. You need to be confident in applying the word mass transport. Not so much knowing what it means, but recognising that if a lot of something is moved at a time, it's going to be a form of mass transport. So we're going to be talking about it in terms of insects and fish, and then we're going to be looking at the heart and the lungs, and then finally at plants. And all of those are mass transport systems. And the idea of mass transport is it helps to speed up the process of gas exchange in the organism that it's happening in. And the final adaptation that we're going to take a look at is how the tracheoles and the fact that they're covered with water, with that body fluid at the end of them, is actually beneficial to gas exchange. So at rest, the end of the tracheoles, and we'll do another rubbish diagram, are covered in that body fluid. We've spoken about this a bajillion times so far this lesson. And obviously those body fluids have got to come into contact with some cells. Maybe these are muscle cells to help the insect move. We'll use an example of a locust, perhaps. And in those cells, they're going to produce lactate. So I'm just going to do little blue lactate particles in our cells. Lactate molecules. And these lactate molecules are able to dissolve. Okay, so what they can do is they can dissolve in the cytoplasm and make a really acid-rich situation, lactic acid. Okay, so we don't want our cells to become acidic. But the dissolving of lactate to make that acidic cell cytoplasm means that we're going to get a reduced water potential in these cells. So they've got a lower water potential, and remember that a lower water potential means a more negative water potential. But what that means is that all of the fluid that's covering the end of the tracheoles is going to want to move by osmosis into the cells. And in doing that, what actually happens is that the volume of the water that's found at the ends, now that it's migrated away, it's moved via osmosis away from the tracheal ends, it sort of increases the space that we've got there to draw air in, like a sucking motion, like a straw. And this is a mechanism that, again, helps transfer gases in the insect. And now the final things we're going to look at are the spiracles. So we've spoken previously in this lesson about the fact that the spiracles are able to be opened and closed. And actually they are open and closing by a valve. So remember, they can be closed as in there's no pore, or they can be open where we've got that gap or that pore in the centre. And as you can see, we've got a highly folded surface, some kind of infoldings of that membrane on the inside of the spiracle at the start of the tracheoles that hopefully will help to increase the surface area. But what do we think the issue is if the spiracles are opened all the time? Why is it important that they can open and close? And while you're thinking about this, also consider what would happen if we were to breathe with our mouth open all the time. Think about how that would feel. What would that do to us? So pause and then play again when you're ready. Now, if you think about us with our mouths open, it's the same for the spiracles. You're going to end up losing water vapour and the organism is going to end up dehydrating. So the spiracles are kept closed generally to minimise water loss. We generally walk around in our day-to-day lives with our mouths closed and only really breathe with our mouths if we need a rapid oxygen intake. Okay, and these spiracles are similar in that they only open periodically to allow that gas exchange in and out. So they're not open all the time, going to get dehydrated and no one needs a dry tracheal. For the last few slides of this lesson, you guys are going to do some independent work. So you're going to have a go at the How Science Works Spiracles Movement Task on page 134 of the Blue Oxford book. And you can pause now, and when you're ready to play again, I'll just go through the answers. I haven't got the questions with me again. It's not an exam question to work through, so I'll just show you them in a second. And then on the next slide, there's another task, and then we're done. Okay, so pause now, spend some time doing those tasks, and play again when you're ready to see the answers. So these are how your answers should look. Each mark is shown by a 1 in a brackets after the point. Can you please check that you're using your keywords properly? Make sure that you're recognising that the spiracles are either opened or closed. Make sure you're talking about concentration gradients if it's relevant. And then we'll go on to the final slide. So pause here if you need to have a look at the answers. Could you correct them if you've got any of them wrong? And if you've got any questions, let me know. Could you please summarise the final paragraph on page 134? That's the blue book again. And then have a go at those 3 questions. They're independent thinking. I want you to use your knowledge and your own research to have a go at these questions. So why is the reliance of the tracheal system on diffusion an issue in insects? In what other way does being small benefit insects? And are there any insects with partially developed internal exchange systems, such as lungs? Once you've done that, that's the end of the lesson. You can share your ideas with me if you like. Thank you very much for today. I hope you found it useful, and I'll see you next time for our lesson on gas exchange in fish. Bye bye.